Hello, I'm Dr. BCMR, Assistant Professor at Shastra. Today we will discuss something about how the chemical nature of the genetic information is deduced. And out of many of the experiments, the first one that really has thrown open the field of molecular biology, especially that of the genetic information, comes from Griffith's transforming principle. The experiment, Griffith's experiment, and he has made a point called as transforming principle. He has performed several experiments, and in one of the papers that was published in 1928, the significance of pneumococcal types, and in there he published a 47 or 40 page paper in which he describes a wide variety of strains and then the experiments, the ability of the strains to cause diseases in mice, and thereby he has shown that how one strain can change into another strain. Here we will just talk the gist of the experiment, and predominantly these experiments were done using Streptococcus pneumoniae. And then there are two different strains. One of it is the rough strain, and the other one is called the smooth strain, or strain S or strain R. Basically, the difference between them is about the colony morphology. Say if we take a plate, agar plate, nutrient agar plate, and in that we put one bacterium, say we have put say 3 different or 4 different bacteria at different points, and we leave them in incubator for say 12 to 24 hours, and the bacterium would divide and would make something like a visible colony like this. That is called a colony because each of them is dividing by binary fission. One is a copy, exact or is identical in terms of features and morphology with the other, and that is called as one colony. And if we observe these strains, they have differences in the way the colony looks like. Say those that of the S strain looked something, they had a smooth surface like this, whereas the other strains had a rough surface, and based on that they were called as R or S strains. There were also the S strain formed bigger colonies compared to that of the smaller colonies. So the difference between them is because of the capsule that is present. The smooth strain had capsule around it, whereas the rough strain did not have any capsule. So something is missing in this, or S strain has something extra. That is why this is a good model organism at that time to perform several experiments. It is because there is a difference in the way they cause disease in test animals. Say for example, we take a mouse and inject this bacterium into it. There is a probability that this mouse could catch an infection, septicemia, and that could be there is a fight between these two organisms. Say we have put in the bacterium, or we can say as a pathogen. When the pathogen enters into the organism, something like a mouse, the mouse has immune system. The immune system will kill these bacteria and get rid of them, so that thereby protecting the mouse. But there is a difference in the way R strain and S strain cause disease. S strain is successful in causing a disease or an infection, whereas R strain cannot, and that is primarily because of this capsule that is present. The capsule protects the bacterium from the immune system, whereas because the R strain does not have the capsule, it is vulnerable to attack, and then the immune system will clear off the bacteria. thereby protecting the mouse. So these Griffith used these two. One is the Streptococcus pneumoniae and the mouse as the model organisms to test how they are different. And he has noticed that one strain changes to other strain. That is called as, say, in literal meanings of English, you can say it as transformation. In biology, you will get the word transformation several times, and it may mean different. Here, transformation in one case would mean that uptake of DNA, which is usually with regard to prokaryotes, the bacteria. And the other meaning of transformation in biology is with regard to eukaryotes or multicellular organisms, a cell becoming cancerous. That is, the cell is no longer under control of the cell cycle regulation, because of which it will grow and form a cancer. So that is also the word used there is also transformation. Anyway, so we'll go into the experimental experiment, how it is done, what is done. I hope we'll be able to deduce. This is a nice one to use deduction to infer something. So he has taken R strain and then injected into the mice, and so the mouse lives. So what does that say is it says that R strain is not virulent. And then S strain, he has taken S strain and injected into my mouse, and mouse dies. Infection has been established, right? And in the next one, he has taken the smooth strain and then boiled it at different time points. For duration is varied, temperature also could have been varied. If you look into the paper, sometimes it's 65 degrees centigrade for 15 minutes or 1 hour, and many other experiments like that were done in the previous paper that I have shown. But the point is, he has taken the S strain, but then it was heat killed. So there is no live bacteria in there. All there are molecules. It could be lipids, carbohydrates, RNA, DNA, everything else, but no live cells in there. And when that resultant fluid was injected into mouse, a mouse is alive, indicating that the heat-killed S strain cannot induce the disease. So the next thing that was done is something important. We don't exactly know why he has done that, probably. We have to read the whole paper. But that is also an important thing as to why we should be reading those. Because it's about the thought process or the idea of why to do this way, right? The general ideas that he might have had. Now, when he has taken R strain and S strain that is heat killed, we should remember that independently they do not cause any disease, right? That is evident from here and here. And he has given that mixture to mice, and then what he noticed is that the disease was established, that the mouse indicated by the death of the mouse. So, and in all of his experiments, wherever the mouse died, he isolated what kind of bacteria is there. Here he got S-type strains, and in this case he also got S-type strains. That is abnormal, isn't it? Because what was alive is the R strain that is given, and all the S strain that is heat killed was given. So once the mouse died, when the bacteria was isolated, it is S-type strain or S strain. How can that be possible? That is to indicate that R strain has changed to or transformed into S strain. And that is the first time we have seen, or probably an experimental evidence about something becoming something else. And that is what referred to as the transforming principle here. Right? Now we know what may have happened. As we'll see, what may have happened is the R strain has a mutation because of which it cannot synthesize the capsule. This is R strain. Whereas the S strain, it has the functional gene for capsule, so it can make this capsule, something like this. Now, when this is killed, all the capsule gene, every other DNA also, would have been like this, and the R strain must have taken up the gene for the capsule. Say this is the capsule gene responsible for encoding enzymes that will produce this capsule, and then the R strain has become S strain, because all the difference between the R strain and S strain is the presence or absence of the capsule, which of course will be because of the capsule gene. And that is what we know now. At that time, he didn't know it. For that time's knowledge and technology. And this is interesting for several reasons. One is the way the experiments, or if we look into the paper, we'll find that there are so many experiments that he has done, and then this is obsessively making more and more experiments. He has designed the experiments very well, and then he has not left anything out of observation. These are some of the things we need to learn from the way these scientists keep thinking about or thinking of. That is about transforming principle discovered by Griffiths. Thank you very much.